Amino acid pool. The entire collection of free amino acids in the body. Essential amino acids. An amino acid that cannot be synthesized by the body and thus must be obtained from the diet. Non-essential amino acid. One of 11 amino acids that are synthesized in the body and are therefore not necessary in the diet. Oxidative deamination. Conversion of an amino acid, NH group, to an alpha-keto group with removal of NH. Reductive amination. Conversion of an alpha-keto acid to an amino acid by reaction with NH. Transamination. The interchange of the amino group of an amino acid and the keto group of an alpha-keto acid. Urea cycle. The cyclic biochemical pathway that produces urea for excretion. How do amino acids enter the pool? 1. Degradation of cellular proteins. 2. Dietary protein breakdown. 3. De novo synthesis of non-essential amino acids. How do amino acids leave the pool? 1. Protein synthesis. 2. Conversion to other nitrogen-containing CMPDs. 3. Conversion to glucose or fatty acids for energy. How does protein degradation occur? 1. Ubiquitin, proteasome pathway. 2. Lysosomal hydroxylases. What needs to happen to proteins before they can be absorbed in digestion? Converted to amino acids for digestion. What are proteases, generally? Digestive enzymes released in stomach and small intestine by enzymes released from pancreas for digestion. Give 2 examples of proteases and where they are found. 1. Pepsin found in stomach. 2. Trypsin found in small intestine. Describe 3 main steps which nitrogen gets removed from circulation. Where is the nitrogen taken? Nitrogen is taken to the liver by 3 main ways. 1. Transamination, amino group transferred to alpha-ketoglutarate to make glutamate in muscle, and to alanine, normally low ECF but damaged to tissue will make transferases increased. 2. Oxidative deamination, amino group released as ammonia from glutamate in liver and kidney producing alpha-ketoglutarate. 3. Transport of ammonia to the liver, pyruvate is made into alanine. What is the main way the body disposes of amino acid nitrogens? Urea cycle. What is the ultimate result of the nitrogen removal process, specifically the 3 steps to nitrogen removal? Glutamate and alanine transport into the liver. Nitrogen needs to attach to another molecule. Body doesn't want ammonia free-flowing around due to potential toxic effects. Ammonia decreases blood pH. Increases equals more alkaline. Describe the role of L-citrulline in the urea cycle. In mitochondria, carbonyl phosphate Krebs intermediates make L-citrulline, which then leaves the mitochondria. In the cytoplasm, it combines with aspartate, which goes through urea cycle to release N group as urea. Describe the fate of carbon skeletons of amino acids. 1. Can go into Krebs cycle. 2. Can enter glycolysis equals alanine pyruvate. Differentiate glucogenic versus ketogenic. Glucogenic amino acids that can enter as a Krebs glycolysis intermediate, leucine and lysine, are ketogenic. Leucine and lysine are ketogenic, while all other amino acids are glucogenic. True. Amino acids can be ketogenic and glucogenic. True. Differentiate between non-essential and essential amino acids. Non-essential equals can be synthesized, essential equals need to be consumed to use. The synthesis of amino acids come from where? Intermediates from Krebs glycolysis are synthesized from another amino acid. Name 2 amino acid synthesis disorders common in humans. 1. PKU equals phenylketonuria. 2. Homocystinuria. Describe in detail the 2 amino acid synthesis disorders common in humans. 1. PKU equals enzyme to catabolize phenylalanine is not functioning, resulting in increased phenylalanine, leading to retardation and hypopigmentation. TX by reducing phenyl in diet, tyrosine becomes essential amino acid. 2. Homocystinuria equals mutation in enzymes that synthesize cysteines, resulting in skeletal abnormalities, osteoporosis, mental retardation, and ectopia lentis. TX by restriction of methionine diet, if not genetic cause, increased B vitamins for coenzymes. How long is insulin half-life outside of beta cells? 6 minutes outside beta cells equals tight temporal control. What are the hormones associated with metabolism? Insulin, glucagon, and epinephrine. Beta cells increase insulin production with increased serum glucose, and beta cells decrease production of glucagon with increased serum glucose. False. Beta cells secrete insulin, alpha cells release glucagon. F. Norepinephrine will inhibit release of insulin. False. Epinephrine inhibits release of insulin. Describe what will happen to the following levels of each if insulin is released. 1. Glycogen synthesis activity. 2. Glycogen phosphorylase activity. 3. Blood transporters in muscle and adipose tissue. 4. Fatty acid degradation via HSL. 5. Protein synthesis. 1. Increased glycogen synthesis activity. 2. Decreased glycogen phosphorylase activity. 3. Increased blood transporters in muscle and adipose tissue. 4. Decreased fatty acid degradation via HSL. 5. Increased protein synthesis. Describe specifically what insulin does on the effects of metabolism. 1. Works by binding to receptors on surface of most cells. 2. Receptors are tyrosine kinases. 3. Receptors dimerize and kinase activity give signal cascade. 4. Leads to phosphorylation of IRS, changing metabolic activity through enzyme activity and gene expression. Glucagon release will trigger what two pathways? Glycogenolysis and gluconeogenesis. Essentially, glucagon promotes release of energy stores. Describe what happens with glucagon release. Binds to receptor activating adenyl cyclase, which increases CAMP greater than pKa activation greater than changes enzyme functions. Describe the effect on the following when glucagon is released. 1. Glycogenolysis. 2. Amino acid uptake in liver. 3. Lipolysis. 4. Ketogenesis. 5. Glycogenesis. 1. Increase glycogenolysis. 2. Increase amino acid uptake in liver. 3. Increase lipolysis. 4. Increase ketogenesis. 5. F. Epinephrine supports glucagon signaling. True. F. Liver, kidney, and muscle cells have glucagon receptors. False. Muscle cells do not have glucagon receptors. Why does ETOH affect gluconeogenesis? Increased lactate formation and ETOH metabolism increases NADH. A blank hypoglycemic response occurs when there is a slow drop in glucose concentration due to epinephrine not being involved. What are the symptoms of this? Neuroglycopenic response, confusion, slurred speech, coma. A blank hypoglycemic response occurs with a rapid decrease in blood sugar. What are these symptoms? Adrenergic hypoglycemic, anxiety, palpitations, tremor, and can also get neuroglycopenic symptoms. What are 5 similarities among replication, transcription, and translation? Necessity of large protein complexes, accessory factors, high fidelity, complementary base pairing, and endergonic, require energy input. The genetic code contains no reading frame. Amino acid codons are written in this direction. 5 to 3. Anticodons are written in this direction. 3 to 5. The 3-step codons are UAG, UAA, and UGA. The start codon is August. Codons with second position pyrimidines encode mostly hydrophobic amino acids. Codons with second position purines encode mostly polar amino acids. Change in the first codon position tend to signify similar amino acids. The alignment of two RNA segments is antiparallel. The codon in mRNA base pairs with the anticodon in tRNA via hydrogen bonding. This enzyme ensures that the correct amino acid is selected for covalent attachment to tRNA. Aminoacyl tRNA synthetase. Attachment of amino acids to tRNA requires ATP. The overall reaction of amino acid attachment to tRNA is driven by hydrolysis of inorganic phosphate generated in an initial reaction. Clockwise from the top, the four arms of a tRNA are amino acid arm, TWC arm, anticodon arm, D arm. The 3' end of a tRNA contains adenine, aminoacyl group. When the 5' anticodon base is U, the 3' codon base could be A or G. When the 5' anticodon base is G, the 3' codon base could be U or C. When the 5' anticodon base is I, the 3' codon base could be U, C. Hooray! The 5 important functions of the ribosome are mRNA binding for high-fidelity codon readings, tRNA bindings, translation mediation, peptide bond formation, movement. Most of a ribosome's mass is RNA. Ribosomal proteins consist of a tail with these properties. Basic residue-rich, positively charged. The function of ribosomal tails is neutralize repulsive RNA segments. The A site binds AAtRNA. The P site binds peptidyl tRNA. The E site binds decylated tRNA. Direction of polypeptide synthesis. NC terminus. Peptidyl transferase activity adds an incoming amino acid to a growing proteins. C-terminus. Ribosomes read mRNA in this direction. 5-3. How is the initiation site recognized? Initiation factors. Base pairing between blank and blank helps protect the translation initiation site. mRNA, a pure enriched sequence centered 10 nt upstream of the start codon, complementary to 1 in the 16S. rRNA. Shine-Dalgarno sequence. This initiation factor assists with F3 binding. If 1. This initiation factor binds initiator tRNA and GTP. If 2. This initiation factor releases mRNA and tRNA from recycled 30S subunit and aids new mRNA binding. If 3. This elongation factor binds amino acid tRNA and GTP. EF2. This elongation factor displaces GDP from EF2. EFTs. This elongation factor functions in GTP binding and hydrolysis. EFG. This termination factor recognizes UAA and UAG stop codons. RF1. This termination factor recognizes UAA and UGA stop codons. RF2. This termination factor stimulates RF1, to release via GTP hydrolysis. RF3. This termination factor combines with the FG to induce a ribosomal dissociation to small and large subunits. RRF. The 3 stages of chain elongation. Decoding, transpeptidation, translocation. In this step of chain elongation, the ribosome selects and binds an amino acyl tRNA whose anticodon is complementary to the mRNA codon in the A site. Decoding. In this step of chain elongation, the peptidyl group in the P site tRNA is transferred to the amino acyl group in the A site. Transpeptidation. In this step of chain elongation, the A site and P site are respectively transferred to the P and E sites, along with their bound mRNA. Translocation. The process of chain elongation is powered by hydrolysis of GTP. Any protein that hydrolyzes and dissociates GTP is a G protein. The most abundant protein in the E. coli cell. EF2. The proofreading for accurate tRNA selection is powered by GTP hydrolysis. The ribosomal transpeptidase reaction is catalyzed solely by RNA. Translocation is powered by EFG and GTP hydrolysis. Because the binding sites for these two proteins on the ribosome overlap, their binding is mutually exclusive. EFG and EF2. In eukaryotes, termination codons are recognized by ERF1. Proteins may undergo post-translational modifications including proteolysis, covalent modification, translocation,